Okay, sup nerds, it's Lindsay again. So today what I'm going to do is I'm going to be going over some of the core biology that tends to be high yield on the MCAT because that's the stuff I've been studying a lot. I started out with behavioral sciences and now I'm moving into biology, and then I'm going to from there move into general chemistry, then probably organic chemistry from there, then physics and math, and then probably be doing biochemistry last. Because that's the hardest subject for me, so I want to make sure I review all the things that inform it first, like the biology and the chemistry. So I'm taking some of this stuff from the MCAT quick sheets that came from the Kaplan. I went ahead and bought their whole pack with the different books and the quick sheets in it. I think these are really wonderful, these quick sheets, if you can get your hands on these. It's not, of course, it's not thorough. That's not what it's meant to be. It's quick sheets. It's just the very, very basics, kind of almost tasters of what tends to be the high yield science. So right now I'm going to go over the cell and reproduction and just go over everything and kind of share some of my little ways that I come up with for remembering things and for figuring things out. And hopefully it'll be beneficial to someone. If not, doing this is still beneficial to me, so it's still a win for me in my book, even if no one else gets anything out of it. But anyway, let's go ahead and get into it. So starting with cells, anyone who passed high school biology knows that when it comes to cells, there's prokaryotes and there's eukaryotes. So there are definitely a lot of differences between them, but for our purposes, for MCAT purposes, really the important difference is that the eukaryotes have a nucleus and the prokaryotes do not. They have that little kind of bundle of nucleoid of that genetic material. So the eukaryotes are the one that have the nucleus. So the picture of the eukaryotic cell, it doesn't have it in the quick sheets. I kind of wish it does, but of course I couldn't put everything in there. But if you just Google a eukaryotic cell, you'll find something that looks sort of like this. This is something that we're probably all familiar with. We've all seen this in a textbook. We've probably at some point in our life all had to make a diagram of this. I know I did back when I was in school. So back then, when we were younger, we basically just had to name all the different organelles in it. We had to name what they all were and maybe know a little bit about what they do. For when it comes to the MCAT level, it's not so much about just being able to name nucleus, mitochondrion, lysosome, smooth endoplasmic reticulum, rough endoplasmic reticulum. It's about knowing what they do. So the way I think of approaching it is in each of those organelles, what goes on inside that organelle? What sort of things are happening inside there? That's the way I like to think about it. What's going on in there? Starting with the nucleus. Nucleus, of course, is something that is unique to the eukaryotic cells, and that's where it has all its genetic material. That's where it has its DNA. And the prokaryote does not have that. It has, find a picture of the prokaryote, it has this little mess right here, that kind of bundle of genetic material right there that's not encased in anything, that nucleoid. So it has the nucleus, also has the mitochondrion, which once you get into higher levels of biology and into biochemistry, ooh, that mitochondrion is gonna come back for you and bite you in the ass. That's where so many important things happen. It's where you have the pyruvate dehydrogenase. It's where you have the lovely, our good old friend, the citric acid cycle, or Krebs, or TCA, whatever you've heard it called. Those are all different names for the same thing, but that's where that lovely guy is. You eventually have to just learn to love and embrace the citric acid cycle. I know you start out just hating it because it's so complicated, but eventually you gotta just learn to love it. As far as mnemonic devices for coming up with it, you don't necessarily need a mnemonic device for the Krebs cycle. I know a lot of people find them to be very helpful to remember the different enzymes in it, the different stages of it, but some people I know just did it through pure brute memorization. A really good friend of mine, actually, he just through brute memorization remembers the citric acid cycle. But if you're still looking for a mnemonic device for it, the one that it's a really strange one, but for whatever reason, the one that stuck with me was on California's Isle, kids scorn straight-faced men. I don't know why that has stuck with me so much, but that's starting with oxaloacetate and then going around to the malate. And for whatever reason, that just got in here and has not left. And that's how I've that's how I've remembered the stages of the citric acid cycle. And of course, it's not just all about knowing the stages. You also need to know what else happens in that cycle, but we won't get into that right now. That's more like biochemistry. Right now we're trying to talk more about just basic biology. It's also going back to the mitochondria. That's not just where the citric acid cycle is, it's also where the electric transport chain is. It's where we have the oxidative phosphorylation, which is also pretty complicated, a thing people have some trouble with. It's where you have your beta oxidation, you have some gluconeogenesis there, and it's also where you produce ATP. Very, very important, which is why you've probably heard that old joke that mitochondria is the powerhouse of the cell, and that's really why, because that's where we produce ATP, and ATP is really energy currency. A thing I found out about me actually pretty recently, I only found this out a couple years ago, but I found out that I actually have a genetic disorder where my cells, inside all my teeny tiny little cells, and then inside that in the even tinier mitochondria, my cells only produce ATP at about 20% the normal level, which it turned out was why for my whole life I was having a lot of chronic fatigue and not having pretty low energy. So that's sort of an interesting thing about that just really illustrates why that kind of old joke is true, that mitochondria really is the powerhouse of the cell, because my mitochondria still aren't powering out what at the level they should be. That was why I was tired all the time. So lysosomes, for remembering what lysosomes do, this I think is one of those times where it becomes helpful to know about word roots for Greek and Latin word roots. Lys, L-Y-S, that basically means like decompose or break down, something along those lines. And the lysosome is basically where things get broken down. They just kind of suck things up into them like a little vampire, suck things up, and they lyse them, they break them down. So that's one of the times I think it's really helpful to know word roots. Rough endoplasmic reticulum, smooth endoplasmic reticulum. The big differences between the two are obviously one's rough and one's smooth. The rough is the one that is studded with ribosomes, but beyond that, rough endoplasmic reticulum is where we synthesize proteins for the most part, and then the smooth endoplasmic reticulum is where we synthesize lipids. So there's a few other things as well, but that's like in general the big difference between them. The Golgi apparatus, that's where we have some modification of those proteins. Peroxisomes. Once again, breaking down those words, very helpful. Think about that oxy inside the peroxisomes, where you can see, even in my notes, I underlined the oxy because that's they contain hydrogen peroxide, and then they're the site of the beta oxidation. So oxy, very helpful for that, right there in the word. This diagram I think is really wonderful. I'm glad they have this in the quick sheets, this phospholipid bilayer. It's really good, important to remember the structure of that, because it's the reason why it looks like that is very important. It's because these guys have their little heads up here that are hydrophilic. If we pretend that on the outside this is all full of water, it's not literally just full of water, but if you are thinking there's water around here, these guys are hydrophilic. They want that water, they want to be touching that water, and then their tails down in here are hydrophobic. So they cluster together like this, really, really tight like that, and form this layer, this bilayer. Bi means two, there's two layers right here, where those hydrophilic heads can be exposed to all that fluid, but then they protect those hydrophobic tails from having to be exposed to the fluid. The cell theory, also very important. Cell theory, I think, is the thing you typically learn on like the very first day of biology, but just really make sure you do know it. The three original tenets of cell theory are: all living things are composed of cells. The cell is the basic functional unit of life, and that cells only arise from pre-existing cells. I remember on my diagnostic test for Kaplan, they did have a question in there where they like threw in a red herring where one of the options was like cells can arise from existing cells and inorganic matter, and of course that's incorrect. They can only arise from pre-existing cells. So those are the three kind of classical tenets, but then recently we've had this fourth tenet that they've added as well as we've learned more. Basically, that's kind of the nature of science. We're kind of always learning new things, discovering new things, and as we've made some advancements, we've added this kind of fourth tenet to cell theory, that cells carry genetic information in the form of DNA, and that this genetic material is passed from parent cell to daughter cell. So that's something that may come up. It may be relevant that you need to know that fourth tenet as well. Moving on to prokaryotes, much simpler, as you are aware. When it comes to prokaryotes, there's two types, bacteria and archaea. I believe it was once thought that archaea are a type of bacteria, that for a long time people thought that they were just a kind of bacteria, but once again, as we've learned more, as technology has advanced, we've been able to find out more, we've actually found that archaea are a lot more unique to themselves than we had once thought, than we originally thought. But bacteria and archaea are both prokaryotes, which typically look a little something like this. They're not always this shape though. This one, the kind of cylinder shape here, is what you would call like a bacillic kind of prokaryote, the way it's shaped like that. There's also round ones, spherical ones that are called cocci or coccic cells, and then they also have ones that are like a sort of spiral shape, and then those are called spirally. So that's pretty easy to remember. Not a whole lot you need to know about prokaryotes, I would imagine, for the MCAT. I don't remember there being a lot of questions regarding prokaryotes on there, besides just knowing the difference between gram-positive and gram-negative bacteria. That's also here in the quick sheets. Difference between gram-positive and gram-negative. Gram-positive bacteria have large quantities of peptidoglycan in the cell wall, while gram-negative bacteria have much smaller quantities of peptidoglycan with lipopolysaccharides. So that's the difference right there. That's something that would be probably important to know. And that also that prokaryotes divide by binary fission. That means they basically just split in half, and that's how they reproduce. They split right along that midline and form those daughter cells. Other than that, I think not a lot of stuff regarding prokaryotes is considered high yield as much as stuff concerning eukaryotes. Moving on to reproduction, I'll talk about this a little bit and then give everyone a bit of a break, and I'll just go back to studying on my own. So when it comes to cell division, two different kinds of cell division, mitosis and meiosis. They're very similar. Once you basically understand one, you pretty much can understand the other because they're very similar. I think this is a really great diagram that they have in the quick sheets where it shows the mitosis and the meiosis. So basically we start with interphase, and then we go into prophase, metaphase. This is all the same in mitosis and meiosis. Interphase, prophase, metaphase, anaphase, telophase. Then in mitosis, from there, we just go into the two daughter cells, and then we're done. DUN, done. Then when it comes to meiosis, we're not quite done yet. We have those daughter cells, but then we kind of start over. Then we go back into the metaphase, then anaphase, then telophase, and we end up with 4 gametes at the end that have half the number of chromosomes that the original cell had. This acronym was actually one that I had been taught before, the PMAT, prophase, metaphase, anaphase, telophase. That can help you to remember that order that they go in, so I think that's very helpful. Then the important difference to remember between those two, where they occur basically. Basically, meiosis only occurs in sex cells. It's only in spermatogenesis, basically sperm cells, or oogenesis, egg cells. That's where we have the meiosis, and that's why they have half the number of chromosomes, because then they will come together and form a full cell with a full number of chromosomes. Mitosis, we end up with 2 cells that are exactly like the parent cell up here. So these daughter cells are identical to this parent cell up here. That's the kind of stuff where basically everything else, anything that's not meiosis is mitosis. When you hurt yourself, you cut yourself, your skin heals over by mitosis, by your cells dividing and creating more skin to heal over it. When it comes to early development, we start with the cleavage of the cell, which is those mitotic divisions. Then we have the implantation, where the embryo implants during the blastula phase. Then we have gastrulation, where we form the ectoderm, the endoderm, and the mesoderm. And then neurulation, where we actually develop the nervous system. So this was also something that I had been taught before, these different ways of remembering the difference between ectoderm, endoderm, and mesoderm. So for ectoderm, I have been taught this, think about a tractoderm. The ectoderm forms your nervous system, your epidermis, your lens of your eye, and your inner ear. So we think about things that make you attracted to another person, how you feel when you're attracted to another person. If you see someone that you like, you see them with your eyes, you hear them with your ears. Maybe, you know, in your epidermis, you start getting some little goose pimples or something like that because you're attracted to that person. And maybe that gets your nervous system activated a little bit if you're attracted to another person. Endoderm, we think about the endernal organs. Bear with me, endernal organs, I know. So that's the lining of the digestive tract, the lungs, the liver, and the pancreas, those internal organs for the endoderm. Then when it comes to the mesoderm, the way we think about that, we think about it as the means-oderm. The mesoderm turns into your muscles, your skeleton, your circulatory system, your gonads, and your kidney. So basically, muscles and skeleton, those are your means of moving around. That's your means of moving your body throughout space. Your circulatory system, that's your means of moving things throughout your body, of moving blood and oxygen and all those other things you need throughout your body. Your gonads, that's your means of reproduction, correct? Even your kidney, that's your means of filtering out your body, of filtering things that you don't want out of your body. I know, those last ones end up being a little reachy, but you do what you gotta do to remember stuff, I guess. That's all I got so far. I hope this was helpful to somebody. I'll probably do some more studying later today and probably do another ASMR video. I was also probably going to go see the new Happy Death Day today. You know, I love horror movies, so, and I actually really liked the original, so I'll probably like that one as well. So probably going to go see that today and maybe do a review on it later. Other than that, hope the rest of y'all have a good day and, you know, happy studying.